morning to all of you. Today we are going to discuss the life cycle of bryophytes. In our previous lecture we have discussed about bryophytes and their introduction. So in order to explain life cycle of bryophytes, we would take example of liverworts, the life cycle of liverworts, and then the life cycle in overall bryophytes. So let's talk about the life cycle in liverworts. It begins with gametophytic phase in liverworts. So both of these are male and female gametophytes. Here you can see this is a female thalloid body or female thallus, which is haploid. This is female thalloid body, and these finger-like things are rhizoids actually. These are rhizoids in order to actually provide, you can say, a substratum. They actually function as a hook or attachment with the substratum. So these are rhizoids in both the cases of male and female gametophytic body. So this is a thalloid body on which this archegonium is present. This is a archegonium stalk, and this is a archegonium, and from this archegonium female gametophyte is developed, and here this is a male gametophytic body, which is also a thalloid body and haploid, and from this thalloid body this antheridium is developed, and from this antheridium male gamete is developed. So from this archegonium egg is developed, which is also haploid, and the structure of egg, and it is a micropyle. These are the protective coverings around the egg, and from this antheridium this sperm is developed, or male gamete is developed, which is also haploid. Okay, so both of these gametophytic bodies, or both of these bodies are haploid and are thalloid structures. Why thalloid? Because they are not differentiated into true stem and leaves. So after producing egg and sperm from archegonium and antheridium, now they would undergo fusion or fertilization. Here you can see, and this is a oogamous type of actually reproduction. Why? Because only sperm is motile in nature, not the egg. So only sperm is motile, that's why it is oogamous type of fusion. So after the fusion, what would happen? You can see over here, this is a fertilization. This is an archegonium structure, stalk, and this is egg, and this now this sperm is going to enter inside this egg through this micropyle region. Okay, so now here fertilization is taking place, and as you know, when fertilization takes place, zygote is formed, and zygote is diploid structure, because zygote is made up of, or it is developed from two haploid bodies, male gamete and female gamete, and the fusion of both of the male and female gametes produces a zygote, which is diploid. So here you can see the zygote after the fusion of male and female gamete, and oogamous type of fusion, because only sperm is motile, not the egg. So zygote is now has been formed. This is a base, and this is a capsule, a protective covering around the zygote. Now this zygote would form a sporophytic or sporophyte or sporophytic body or generation. This is the structure, and here you can see in this sporophytic body, these spores now start developing after the process of meiosis. So when the meiosis would occur, these spores now will be haploid in nature. So inside this structure, this is a base in order to provide actually a stability or a kind of skeletal system to this sac-like structure. And inside this, these are the elaters. Yellow color, these are elaters. Elaters are actually known as moisture sensitive teeth. The function of elater is actually in the dehiscence of spores. Okay, means in the release of spore from this sac-like structure when they get matured. So function of elater in the dehiscence of spores from this sac-like structure. So after meiosis they would become spores which are haploid. So after meiosis they would become spores which are haploid, and from these spores gametophytic body is developed. Means both male and female gametophytic bodies are developed. So it is all about the life cycle of liverworts. Begins with the gametophytic bodies, archegonium and antheridium. They would develop male and female gametes, means egg and sperm, and the fusion is oogamous type because only sperm is motile. This egg is stationary, so that's why oogamous type of fusion. After fusion, zygotic formation. Zygote is diploid, as you know, and then sporophytic body. Then after meiosis, meiospores or spores are formed, which are haploid, and from spores, both male and female gametophyte are developed. So all about these the life cycle in liverworts, and here you can see spore, as we have studied earlier in our previous lecture. The sporophyte is dependent on a gametophyte, although gametophyte is predominant phase in the life cycle of bryophytes, and the life cycle is known as haplodiplontic. I told you, sorry, I will tell you later. And then both gametophytic. Why do we call them haplodiplontic? Because both gametophytic and sporophytic phases are multicellular and are free living. So here you can see, and this diagram you would see at the last pages of the NCERT of this very chapter. So this is a life cycle. This is, you can say, outline of the life cycle in bryophytes. Here you can see this sporophyte, which is diploid, and this sporophyte would undergo meiosis to form spores. And when meiosis would occur, reductional division, diploid sporophyte now would become haploid spores, and these haploid spores uh, would be developed into gametophyte, haploid gametophyte, and this haploid gametophyte means both male and female gametophyte. Both of male and female haploid gametophyte would form male gamete and female gamete. Hence, gametogenesis would occur, means formation of male and female gamete, sperm and egg. And then syngamy means a fusion would take place in order to form zygote, which is diploid, and from zygote again sporophyte, again undergo meiosis, spores. So this would carry on again and again in continuity. And here you can see we have divided the two sections. This upper region or this upper section is all about diploid structures, zygote, sporophyte, diploid structures, and these spores and gametophytic bodies, as they are haploid, so we have written haploid over here. So actually this life cycle is categorized in haploid phase and diploid phase. So that's why haplodiplontic life cycle in case of bryophytes. And why do we write haplo first then diplontic at last? The reason is haplontic or haploid phase is long lived as compared to diploid or diplontic phase. That's why we write haplodiplontic life cycle, because both haploid and diploid phase are free living, and they do form very prominent, actually they would form very prominent their importance in the life cycle of the bryophytes. No doubt, gametophytic body or gametophytic phase is dominant, and sporophytic phase is dependent on gametophytic phase for stability and for water. But both of the phases are, you can say, they are free living, and uh, both have very prominent place in the life cycle of bryophytes. That's why haplodiplontic. Why haplo first? Because haplontic phase is long-lived as compared to diplontic phase. It's all about life cycle of bryophytes. In the next lecture, we would start with pteridophytes. Thank you.